Hello friends, I'm Mayantika and in this video I will be discussing about the vegetative reproduction of Chara. Now Chara is a very important algae because it is part of both BSc and MSc syllabus. I have made a earlier video about Chara part 1, where I have dealt with the taxonomic importance of Chara, the thallus organization, as well as the ecological, economic and research importance of Chara. Here in this video, I will be discussing about the vegetative reproduction structure, but in the earlier video, I have discussed all those things. So before watching this video, I would request you to go and watch that video fast because you will be getting a basic knowledge about the Chara before we start the vegetative reproduction. So these are the vegetative reproductive structure of Chara. Before I go to the vegetative reproductive structure of Chara, I just say that Chara is a very important algae so far as the ecological importance, because first though it helps in the nitrogen fixation in the aquatic system, because most of the Chara, they have cyanobacteria growing on them epiphytically. So it is a basic framework on which nitrogen fixing organisms grow. So apart from photosynthesizing in the aquatic environment and providing oxygen, it also serves as a very basic structure on which the cyanobacteria can generate nitrogen and enrich the water ecosystem. Apart from this too, that is oxygen generation and nitrogen fixation, this Chara is also very important because it is a basic food source for aquatic invertebrates like snail, shrimp, as well as aquatic vertebrates like the fishes, like the forage fishes, the trout, the blue-gill fish, the various type of bass fish. So this is the third most. It is a basic primary producer in the ecosystem, and on which the many economically important fishes, that is fishes on which we feed, those fishes have get their food from this Chara. So this is the third most important point, and the most important is that the Chara, it stabilizes the sediment on which it is grows, and it cleans the aquatic environment. So these are the fourfold most important reason why Chara is important. And now let us deal with the vegetative strategy. Here we see that this is the stem and this is the root bulbil. So one of the vegetative structure of Chara is that it grows by this bulbil, and this is the amorphous bulbil and this is the Amylon starch. Now, as we know that Chara, it grows over a wide range of climatic condition, that is, it is cosmopolitan in distribution, that is, it is found from 69° north to 49° south. So we see that apart from the circumpolar region, Chara is very much present cosmopolitan throughout the world. So naturally it should be very resilient, and Chara can grow in very low nutrient oligotrophic to mesotrophic water. It can grow in hard water, it can grow in alkaline water, and it is also tolerant to a bit of salinity also. So any single algae which has got so much resilient property should have certain mechanism, certain perinating structures, so that they can tide over the unfavorable climatic condition, because we know that the climatic condition, it is not uniform. Sometimes there is much water and sometimes there is water shortage. Sometimes there is high temperature alternating with the low temperature. So Chara and any organism which are very resilient to such conditions will have certain perinating structures that will help that algae to tide over that particular harsh environmental condition. Earlier, in my videos, in nostoc, chlamydomonas, in the eulothrix, oedogonium, chlorella, all this we have seen that they have got, all the algae, they have got certain form of perinating structures. So here in Chara also we see the perinating structures. These are the structures where by which the Chara, it remain dormant in this structure and they germinate on return of favorable condition. So these perinating structure are the vegetative structures. These are the bulbils, the amorphous bulbils, and the amylon starch. Apart from it, there is also another structure called the secondary protonema. We are going to discuss all these four reproductive strategy, vegetative reproductive strategy in detail in the next slide. So this is the we are going to discuss about the bulbil. In the earlier video we have seen that bulbils are present either at the nodes of the stem or the root. These are the bulbils. They are either present at the rhizoid, that is the rhizoidal portion of the Chara, or at the stem. In the stem also it is present at the base, that is the basal nodes of the Chara. Rhizoidal bulbil are present in Chara aspera species and the stem bulbil is present in Baltica. Mind it that both this stem and the rhizoidal bulbil, they are present at the nodes. These bulbils, they are oval or spherical body and they develop at the stem or root nodes. And when the condition becomes very dry, the entire plant body dies. The Chara, this bulbil of Chara, they are liberated in the ground where they stay dormant for certain time till the condition suitable for germination reappears. When the conditions are conducive for germination, then only it germinates into a new plant. Now is the secondary protonema. Now the secondary protonema are the structures that are developed from the primary protonema. So say for example this is the Chara plant and these are the terminal nodes, these are the intermediary nodes, and these are the intermediary nodes, and this is the basal node. So from as the condition becomes very dry, and these are the rhizoids. These are the rhizoids. So as the temperature dries up, so the upper portion, this upper portion, it dies out, and the lower portion somehow remains. So this is the basal internode, and this is the basal node. So from this nodal region of the older part of the Chara, or say for example the primary protonema, or the older portion of this existing old plant, from the basal node arises the secondary protonema. So this secondary protonema, it looks like something like this one. This is just a thread-like structure which is composed of chain of cells. Here we see that there are chain of cells or thread-like structure. These are the secondary protonema. They are very much looking like the primary protonema. The primary protonema develops into a Chara plant. The primary protonema, it develops into a Chara plant, and from the basal node, this basal node of the Chara plant, old Chara plant, arises this secondary protonema. Now this is the secondary protonema. This entire structure is called secondary protonema. So secondary protonema looks very much similar to the primary protonema and arises from the basal nodes of the old surviving plant. Then is the amorphous bulbils. Now, amorphous bulbils, just like other bulbils, normal bulbil, they develop from the nodes of either the stem or the root of a Chara plant. But this is the normal bulbil. But the amorphous bulbil, they will be of much smaller size and they will be of numerous in number. They will be numerous in number but very small size. Say this is the normal bulbil and this is the amorphous bulbil. So they can arise from the nodes of either the stem or the root of a Chara plant. In the Chara Delhi calculator, it occurs in the stem nodes, and in the Chara fagifera and Chara baltica, it occurs in the rhizoid nodes. Now they are much smaller size than the bulbil, and these small cells, they store all the reserve food matter and they are aggregated at the nodes. They are called the amorphous bulbils. So just like this amorphous bulbils also, just like the bulbils, when they get detached, they either remain dormant during the unfavorable condition, but if the condition is favorable, then what it does, it germinates into a new plant. Remember one thing, that both the bulbil, the amorphous bulbil, protonema and the amylon starch, they are the structures which have abundance of nutrients. That is, in case of Chara, it is the starch. This is the Amylon Star. Now, Amylon Star, it is multicellular aggregation of cells, and the cells, the multicellular aggregation of cells, now each of these cells, they are rich in starch. They are filled in starch, and where are they located? They are located in the lower nodes of the old plants. So at the basal node, this is the basal node and this is the rhizoids. So at the basal node of the Chara plant, we get this amorphous stars. So this is the basal node. This is the this is the basal node from where the rhizoids appear, and here we can see this type of structures, the star shaped structure. This is not a single cell but an aggregation of cells. Just like in the previous slide, we have seen the amorphous bulbil, which are aggregation, and the amylon starch are also aggregation of this cells. So two type of aggregates we see. First is the amylon starch, another is the amorphous bulbil. So these are the multicellular structures, and in the unicellular structure of the vegetative reproduction, we see the protonema and the bulbil. With this, I come to the end of the vegetative reproduction part of Chara. Next, I will be making video on the sexual reproduction of Chara. In case you haven't seen the earlier video of Chara, you can go to the earlier video to get a better understanding. Thanks for staying with me till the end of the video, and if you like it, share it with your friends. Do subscribe to get the latest update of the videos that I make, and thank you. And if you have any suggestions, you want me to make any videos in any particular topic, you can also write it in the comment section. Thank you.